Welcome back, future doctors. Look, I know the exam pressure is absolutely intense right now. Microbiology can honestly feel like this massive, never-ending list of isolated facts that you just have to blindly memorize. But don't worry, we're not doing that today. We are going to take that mountain of information and transform it into a highly structured, point-by-point explainer of guaranteed test topics. We're focusing strictly on high-yield bacterial morphology to get you organized, confident, and totally exam-ready. Okay, let's dive right into our roadmap for this explainer. First up, we'll tackle appendages and motility. Second, we'll look at bacterial spores. And finally, we'll map out the bacterial growth curve. Having this framework in your head will seriously reduce your cognitive load.
Starting with Layer 1, the exterior. Let's talk about appendages and bacterial motility.
Now, what's really interesting here is how we visually differentiate the types of layers outside the bacterial cell wall. When this protective layer is highly organized, tightly packed around the cell, and doesn't just wash off easily, we call it a capsule. Then you've got the slime layer, which is exactly what it sounds like: diffuse, unorganized, and very loose. And then, when millions of these bacteria embed themselves within a self-produced slime matrix on a solid surface, think of a plastic medical catheter, that whole community forms a biofilm. This distinction is heavily tested, so make sure you've got it down.
So the absolutely crucial point here is knowing what these capsules are actually made of. Why? Because examiners love, and I mean love, testing the exceptions. Notice how almost all bacterial capsules, like your pneumococcus, Klebsiella, or Haemophilus influenza, are polysaccharide in nature, basically sugars. But I need you to flag this massive, high-yield fact in your notes right now. Bacillus anthracis is the major exception. Its capsule is made of polypeptide, specifically glutamate. Lock that in your memory. It is a classic, guaranteed multiple-choice question. Now, how do these bacteria actually get around? Well, flagella are their primary organs of locomotion, and they have different setups. You can have a single polar flagellum at one end, which is called Monotrichous, just like in Vibrio cholerae, or the flagella can be distributed all over the entire cell surface, which is called Peritrichous, like what we see in Salmonella typhi. It's also important to briefly contrast those long, wavy flagella used for movement with the entirely different, rigid structure of a sex pilus extending from the bacteria, which has a totally different job.
And speaking of Pili, let's make a crystal clear side-by-side comparison for your exams. Common Pili, which are also called Fimbriae, are very short and highly numerous. We're talking up to 1,000 per cell. Their sole job is adhesion. They act like little grappling hooks, helping bacteria stick tightly to your epithelial surfaces. Sex Pili, on the other hand, are strictly found in gram-negative bacteria. They are long, thick, tubular, and you only find a few of them, typically just 1 to 10 per cell. Their specific job? Forming a literal tube for bacterial conjugation. That's the direct cell-to-cell transfer of genes.
All right, moving to Layer 2, the bunker. It's time to talk about bacterial spores and extreme survival.
Listen, when nutrients for now and survival is on the line, bacteria build an absolute fortress. To remember these highly resistant spore layers from the inside out, just use the simple mnemonic CCCE. That stands for core, cortex, coat, exosporium. The core is the vault holding the precious DNA. Then comes the thick cortex, followed by the tough coat, and finally the outermost exosporium. It's this exact layered structure, packed with calcium and dicocosinic acid, that makes mature spores almost indestructible against intense heat and routine disinfectants. And this brilliantly illustrates exactly what you'll see in your practical exams under the microscope. Spore positions and shapes are constant for a given bacterial species, which gives you massive clues for identification. Look at the variations. Spores can be central, subterminal, or terminal right at the very end of the bacillus. And check out the shape differences. They can be non-bulging, meaning they're just the same width as the bacteria, like in Bacillus species, or they can be bulging, literally wider than the cell, creating this distended, swollen look. That bulging shape is a classic hallmark of Clostridium species. Because these spores are so incredibly tough, we actually use them to our advantage to test if our hospital sterilization equipment is actually working. These are heavy-hitter facts for your exams, so pay attention. If you want to test the effectiveness of an autoclave, you use a biological indicator containing spores of Geobacillus stereothermophilus. If you need to test a hot air oven, you use spores of Bacillus atrophius. The logic is simple. If those incredibly resistant spores are completely dead after a cycle, you know without a doubt that your equipment is successfully sterilizing everything else. Let's hit our final major topic, Layer 3, the timeline. Let's walk through the bacterial growth curve.
Okay, here is the classic curve. To lock this sequence right into your memory, I want you to use the acronym LLSD for the 4 phases: lag, log, stationary, decline. I also want you to anchor your visual memory to how the 2 lines represented here diverge over time. The line for the total count and the line for the viable count track together perfectly at first. But watch what happens at that final decline phase. The viable count drops sharply downward, right? While the total count line stays completely flat.
Let's make a crystal clear distinction between those 2 metrics because, honestly, this confuses a lot of students. The total count includes absolutely everything, both the dead and the alive cells that you physically observe under a microscope using a counting chamber. That line never goes down because even dead cells are still physically taking up space and getting counted. The viable count, however, measures only the living, multiplying cells, usually calculated using the pour plate method. That's exactly why the viable count plummets in the decline phase. The bacteria are rapidly dying off due to toxic buildup in their environment.
Now let's quickly break down those LLSD phases. Think of this summary as your ultimate exam cheat sheet. In the lag phase, bacteria aren't dividing quite yet. They're just getting ready, building up enzymes, and reaching their maximum size. Then comes the log phase, where explosive exponential division happens. And here's a huge MCQ tip. The log phase is the absolute best time to perform a gram stain. Why? Because the cells are highly biochemically active, smaller, and they take up the stain uniformly. Later, in the stationary phase, nutrients run out. The death rate equals the division rate, and bacteria start producing storage granules, exotoxins, and those tough spores we talked about. Finally, in the decline phase, death simply overtakes division.
Okay, before we wrap up this explainer, let's do a quick active recall check. I'm going to throw a rapid-fire, textbook-derived MCQ at you to test your memory and get you right into that test-ready mindset. Question: which bacterial cell structure is directly involved in cellular respiration? Think back to the internal cytoplasmic matrix anatomy you've studied. Got your answer locked in? The answer is mesosomes. These are highly prominent invaginations of the plasma membrane that actually possess respiratory enzymes. This makes them functionally analogous to the mitochondria that we have in human eukaryotic cells. Kind of amazing, right? If bacteria can hack their own membranes to act like mitochondria, what other complex cellular functions might they be mimicking to survive? Keep reviewing those mnemonics, trust the structure we built today, and I'll catch you in the next explainer.